Very good afternoon to all the EAPCET aspirants. So, here will be a mega session on all the EAPCET previous year questionnaire practice from 3 chapters of plant physiology, photosynthesis, respiration and plant growth and development. Previously, we are already dealt with the EAPCET PYQs of transport in plants, mineral nutrition, and now we are going with these units. Let's get started with photosynthesis in higher plants first. So, look into the first question, which is assertion reason type of question. Take 1 minute of time and answer. No hurry at all. Yeah. Look at the question. Assertion, Cran's anatomy is characteristic feature of C4 plants. True. Cran's anatomy. What is Cran's anatomy? The vascular bundles in C4 plants mesophyll. In the leaf of C4 plants, the vascular bundles are surrounded by many layered bundle sheath cells. Fine, right? It is surrounded by many layered or many cells of bundle sheath cells. These bundle sheath cells are in close proximity with mesophyll cells. Okay, they are in close proximity with mesophyll cells. They are attached via plasmodesmatal connections. Once let us recall about bundle sheath cells. Bundle sheath cells consists of agranule chloroplast. Okay, it does not have grana in it. It has agranule chloroplast. Fine, right? It is consisting of agranule chloroplast and it is abundant with Rubisco enzyme, which is world abundant enzyme, and in bundle sheath cell C3 cycle is conducted. Fine, right? So, assertion is true only. Reason, photorespiration is absent in C4 plants. Exactly. Photorespiration happens only in C3 plants. C4 plants are well adapted with Cran's anatomy, wherein inside mesophyll cells primary carboxylation will happen by the enzyme peptidase, and second time carbon dioxide fixation occurs in bundle sheath cells via Rubisco. So, there is no possibility of photorespiration in C4 plants. But then if you clearly look into assertion and reason is nowhere explaining about Cran's anatomy. Here 2 statements are provided and these 2 statements are correct. But then reason is not correctly explaining the assertion. Clear till here? So, here A and R are true, but reason is not the correct explanation of assertion. Option B is the right answer. Next. Here we go with this. Once look into the explanation also. Assertion is true because bundle sheath cells will have agranule chloroplast. Reason is also true. Photorespiration is effectively absent in C4 plants, but it is not correctly explaining the assertion, and here is the explanation which I have already given to you. Look into the next question, ma. Match the following type of question. You can easily answer it. First stable product of TCA cycle, tricarboxylic acid cycle. First stable product. What is the first reaction that happens in TCA cycle? 4 carbon compound, that is oxaloacetic acid, which is 4 carbon compound. It will be accepting 2 carbon compound, that is acetyl CoA. Fine, right? And it will form the first stable product, that is citric acid by the enzyme citrate synthase. Fine, right? So, here you see first stable product of TCA cycle is citric acid, which is option 4. First stable product of C3 cycle. C3 cycle, the name itself is driven with the first stable product form, which is 3 carbon compound, and that is 3-phosphoglyceric acid. Once let us recall the first step, that is carboxylation of C3 cycle. C3 cycle carboxylation step. If you see, carbon dioxide is accepted by 5 carbon compound, that is RUBP, ribulose bisphosphate. Fine, right, and this is one carbon, carbon dioxide. This is catalyzed, the first step of C3 cycle is catalyzed by rubisco, ribulose bisphosphate carboxylase oxygenase enzyme, right, and here the first stable product formed is 3-phosphoglyceric acid. Here it is 2 molecules of 3-phosphoglyceric acid is formed. So, B will be 2 and first stable product of C4 cycle will be oxaloacetic acid, which is 4 carbon compound. Let us recall C4 cycle as well. In C4 cycle, carbon dioxide is accepted by phosphoenol pyruvate, which is 3 carbon compound, and here carbon dioxide is 1 carbon. This is catalyzed by only carboxylase enzyme, that is pepcase, phosphoenol pyruvate carboxylase enzyme. Fine, right, and this forms out the first stable product, that is oxaloacetic acid, which is 4 carbon compound. Clear till here, ma? And then product of nitrogen fixation. After nitrogen fixation is done, it is fixed into ammonia. Fine, right. So, here if you clearly look into the options, A goes with 4, B with 2 and C with 3 and D with 1. Option C will be the right answer. Any doubts anywhere, ma? Clear? Yeah. And then next question. Yeah. Look at this very simple question. In C4 plants, the first 4 carbon compound is formed by following. First 4 carbon compound is formed. C4 plants, the name is after the formation of first stable product in the first step, that is carboxylation. So, here you see it is with carboxylation of pep. In the previous question also we have learnt the same. So, look at the questions which are from the carboxylation steps of C3, C4 cycles only, 2 to 3 questions you can see, okay, be it with the acceptors or be it with the products or be it with the enzymes. So, here you see carboxylation of pep. Phosphoenol pyruvate is 3 carbon compound, it will accept carbon dioxide forming out the 4 carbon oxaloacetic acid. Fine, right? Next. You can go through the answer also. And then next question, which is multi-statement based question. Multi-statement based. This session is highly useful to all the EAPCET aspirants, where complete questionnaire practice is given out in a single session from plant physiology. Yeah, look at this question now. Which of the following events occur during light reaction while synthesizing glucose in green plants? Or you can, as this is the general formula of carbohydrates, synthesizing carbohydrates in green plants. Look at the options carefully. Dissociation of water in the lumen of thylakoid. This occurs in the light reaction itself. But here the question is the events occurring during light reaction while synthesizing glucose. But this happens inside the lumen, which is photolysis of water. So, this is not associated with synthesizing of carbohydrates. Oxygen released in association with PS1, wrong statement. Oxygen is released in association with photosystem 2. Next, photosystem 2 is associated with water and releasing of electrons, true, right. Next, coenzyme is reduced in association with photosystem 1. Exactly, coenzyme is released in association with PS1 itself. Okay, in PS2 coenzyme is not present, which is situated in thylakoid lamellae. PS1 possess coenzyme. Fine, right? So, these two are correct. In statement A, if you see, photolysis of water occurs in oxygen evolving complex, which is situated in inner thylakoid membrane, inner membrane of thylakoid towards lumen. Fine. So, yes, this is true. That statement will go wrong because it is not in the lumen, and photolysis of water or oxygen release is nowhere connected to PS1, it is connected to PS2. C and D goes correct. So, here what will be the right answer? Option C. Look into the answer also once. Yeah. Occurs in the inner side of the thylakoid membrane, and B is incorrect because it is associated with PS2. C will be correct and D is correct because NADP is reduced at PS1 only by the enzyme CoA. Next, look at this question. How are ATP molecules generated during photosynthesis? How they are generated during photosynthesis? Multi-statement based question again. Due to conformational changes during proton diffusion through F0, first you recall where is F0 present. If this is the thylakoid membrane, let us consider this is the lumen side and this is the stroma side. Proton gradient is generated from lumen to stroma, wherein F0 F1 pump is activated. F0 F1 pump is also known as ATP synthase, where F0 is present or embedded inside the thylakoid membrane and F1 is towards the stroma. So, here you see F0 which is embedded inside the thylakoid membrane, of course it will show the conformational change during proton diffusion. True. By movement of protons to stroma through F0 channel, but here to stroma it is from F1, inside stroma it is F1, inside thylakoid lamella it is F0. By movement of protons to stroma through F1 channel, to stroma it is F1 not F0. By breakdown of proton gradient, it is across the gradient not against the gradient. Okay. The transfer of protons is across the proton gradient not against to it. By organization of ATPase, okay, it is by organization of ATPase in flattened membrane sacs. True, it is in flattened membrane sacs. Those flattened membrane sacs are what? Thylakoids. Fine, right. So, here right answer will be A and D, which is option C. Clear, right? Fine. Yeah. ATP synthesis occurs due to chemiosmosis. Once recall what are the prerequisites for chemiosmosis: membrane, F0 F1 pump, proton gradient and a proton pump. These are the four prerequisites for chemiosmotic hypothesis or chemiosmosis. Proton flow through F0 channel and utilization of proton gradient, these are the main things for the gradient to occur. And here if you see the third option, it is not against the proton concentration, it is across the gradient. Fine, right? Next, look at this question, it is on the energetics of C3 cycle. How many ATP and NADPH molecules are required for fixation of 12 carbon dioxide molecules through Kelvin cycle respectively? How many ATP and NADPH molecules are required for fixation of 12 CO2? Look at here. For fixation of 1 carbon dioxide, it requires 3 ATP, 2 ATP for reduction and 1 ATP for regeneration, and 2 NADPH or 2 NADH it requires at the time of reduction. For 12 carbon dioxide fixations, the products formed are 2 glucose molecules are formed, and it requires 3 into 12, 36 ATP and 2 into 12, that is 24 NADH. Look at the answers carefully. It is NADPH. Yeah. Look at the answer. It is 36 ATP and 24 NADPH, which are required for 12 carbon dioxide to fix, and fixation of 12 carbon dioxide molecules will result in the formation of 2 glucose molecules as the products. Clear? Clear with this question? Any doubt anywhere, ma? Now, look at this question, which is assertion reason type. Clearly you see, in assertion, in C4 plants, photorespiration does not occur, 100% true, and this is second time we are seeing the same statement on photorespiration of C4 plants. In reason you see, it is due to a mechanism that increases carbon dioxide concentration at the rubisco site. There is no rubisco site in the mesophyll cells. In mesophyll cells, the enzyme which is taking care of carboxylation is peptidase, where rubisco acts as predominantly as oxygen is wrong. So, here statement assertion is true and reason is false, because in mesophyll cells it is peptidase is the enzyme which is fixing the carbon dioxide, and there is no point of oxygenase activity of rubisco, because rubisco is present in bundle sheath cells. Bundle sheath cells, these bundle sheath cells are deeply inside the mesophyll cells, where oxygen will not be prevailed. Okay, direct entry of 4 carbon compound into bundle sheath cells, immediately after decarboxylation, carbon dioxide will take over C3 cycle. Fine, right? There is no point of oxygen and oxygenase activity inside the bundle sheath cells. So, here if you see statement A, that is assertion is true and reason will be false. Option C. Clear? Now, next question you look into. Choose the incorrect statement. Statement based question. This is on discoveries. In a single slide itself we have learnt all the discoveries. Look into it and answer carefully, ma. Half-leaf experiments proved oxygen is required. Oxygen is required for photosynthesis. Okay. O2 is required for photosynthesis, which is proved by half-leaf experiment. Is that true? Half-leaf experiment proved oxygen is required for photosynthesis. Wrong. Half-leaf experiment is proved that carbon dioxide is essential for the process of photosynthesis. Next statement B, you see, engine house showed aquatic plants release oxygen in light. True, by the hydrilla experiment. Bell jar experiment indicated plants restore air vitiated by animals. True only. Okay. And then Sachs experiment showed chlorophyll is located in special bodies. Wrong. Okay. Sachs experiment, Von Sachs has proved that glucose is formed at the end of the photosynthesis, and he said chloroplast, inside chloroplast glucose is formed. Okay. There is no point of special bodies in his discovery. So, here what is the right answer, ma? Which are the two correct statements that we can go through? B and once again look into it. Half-leaf experiment proved oxygen is required for photosynthesis. No. Carbon dioxide is essential is what the experiment told. B will be correct. Bell jar experiment indicated plants restore air vitiated by animals, which are released from animals. Sachs experiment showed that chlorophyll is located inside the special bodies. His experiment is to prove glucose is the end product. So, here you can go with B and C. B and C. We can go with option D. Clear? Next question. The movement of electrons from P680 to pheophytin and pheophytin to P700. Look at here carefully. From P680, first let us take two photosystems. Photosystem 2 and photosystem 1. In photosystem 2, first the electrons which are excited to P680, this is uphill movement. From P680 to pheophytin, which is the first electron acceptor, this is downhill. All across plastocyanin, which is the last electron acceptor of PS2, till here it will be downhill movement. From plastocyanin to P700, it will be uphill movement. Again from here to NADP, it is downhill. Now we understood it clearly. So, first it will be downhill. From P680 to pheophytin, it is downhill, and again movement of electrons to P700 is uphill. So, first it is downhill and then uphill movement. Option B is the right answer. Clear, ma? Then look at here, next question. Which of the following statements are false regarding C4 plants? Regarding C4 plants, which of the following statements are false? C4 plants lack photorespiration and they have lesser biomass. Yes, they lack photorespiration and they do have less biomass. Bundle sheath cells contain large number of chloroplasts. No, they have large number of A granule chloroplasts. Okay, and chloroplast number is not maximum when compared to mesophyll cells. Mesophyll cell consists of rubisco enzyme. Wrong, mesophyll cell doesn't have rubisco enzyme, it has peptase enzyme. Primary carbon dioxide acceptor is 4 carbon molecule. True. No, wrong. Primary carbon dioxide acceptor is 3 carbon compound, which is phosphoenol pyruvate. Okay. Bundle sheath cells have large number of chloroplasts true only, but mentioning is clear that should have A granule chloroplast. Fine. So, when you look into all the 4 statements, C and D are completely wrong because it does not have rubisco enzyme in mesophyll cells, and primary carbon dioxide acceptor is 3 carbon compound, not 4 carbon. A and B are true. So, option A is the right answer here, A and B. Clear? Next question. Sorry, he asked us for false statements, right? Yeah, this is more important in statement based question, whether he is asking us to identify correct statements or incorrect statements. So, here incorrect statements will be C and D. It is not mentioned over here. Next. Which statements are false regarding photorespiration? If you look into maximum PYQs from photosynthesis, whole lot of questions are asked from photorespiration itself. Okay. Which statements are false regarding photorespiration? Neither sugar nor ATP synthesis occurs. Yes, there is no amount of energy that is synthesized. Photorespiration occurs in C4 plants. Wrong. It occurs only in C3 plants. 2 phosphoglyceraldehyde molecules are formed from RUBP. Wrong. When oxygen is being accepted by RUBP, the first step, which is 5 carbon compound, and Rubisco performs oxygenase activity here. Okay, it performs oxygenase activity, and then it produces the initial products, which is phosphoglyceric acid, that is 3 carbon compound, and phosphoglycolate, which is 2 carbon compound. In C3 plants, photorespiration reduces the productivity. True. False statements regarding photorespiration will be B and C, which is option C. Clear? Any doubts anywhere? And then next question. Choose the correct statements among the following. Chlorophyll is major pigment responsible for trapping light. Accessory pigments also participate. True. In green plants, water is the hydrogen donor. No, it is the electron donor. Purple sulfur bacteria, the oxidation product is carbon. Look at it carefully. And then option C, in purple sulfur bacteria, oxidation product is not carbon, it will be sulfur. Radioisotope studies proved that water as the source of oxygen release in photosynthesis. True. Water is the source of oxygen release because water is the electron donor. Fine. So, here if you observe, hydrogen and electron, both statements goes correct only. Yeah. So, A, B, D. Option C is the right answer. Clear? Next. Chlorophyll A is the primary and auxiliary pigments also help for it. Water is the hydrogen electron donor in oxygenic photosynthesis. Both terms will be correct. Hydrogen donor or electron donor. Purple sulfur bacteria oxidize H2S2 sulfur, not carbon, it is to sulfur. Radioactive isotope studies showed that oxygen comes from water. Next question. Relative concentration of oxygen and carbon dioxide determines which binds to Rubisco. True. If carbon dioxide and oxygen are in the equal ratio, let us say 1:1, then Rubisco binds to carbon dioxide itself. If it is 1:2, more amount of carbon dioxide is present than, more amount of oxygen is present than carbon dioxide, then Rubisco performs oxygenase activity. Clear ma? And Rubisco has much greater affinity for carbon dioxide than oxygen. Is that correct? Rubisco has greater affinity for oxygen, not for carbon dioxide. So, here A is true and R is false. Rubisco does not have greater affinity for carbon dioxide, it can bind to both, as just we have discussed 1:1 ratio. Match the following type of question on discoveries. Sunlight essential to plant purpose. Sunlight is essential to plant process that purifies air fouled by breathing animals. Bacteria used to detect the sites of oxygen evolution. This is by C.B. Van Niel, which is 6. Okay. And evidence for production of glucose is performed by Von Sachs. Role of air in growth of green plants. Which is sunlight is essential for the process of photosynthesis is said by Jan Ingenhouse. Fine, right? And then role of air in plants growth is done by Bell-Jar's experiment by Joseph Priestley, which is option 3. So 4, 6, 2 and 3, which is option D. Clear? Next question. Assertion, reason type again. Here we go with this assertion. Chromatographic separation of leaf pigments shows leaf contain different pigments. True. Pigments absorb light at specific wavelength and they aid photosynthesis, that help in the process of photosynthesis. Major light pigments are chlorophyll A, chlorophyll B, xanthophylls and carotenoids. So here assertion and reason both are correct, and reason is the correct explanation of A, which is option A. Fine, right? A is the correct answer over here. Clear? The question is being derived from Engelsmann experiment absorption spectra. Again assertion reason type of question from non-cyclic photophosphorylation, which is non-cyclic photophosphorylation. When electrons move through transport chain, protons are transported across the membrane. Fine, right? When electrons are moved through transport chain, protein molecules being accepting the electrons, protons are transported across the membrane. True. The primary electron acceptor transfers electrons to plastoquinone, enabling the proton translocation. Once look at that reason point. Fine. How the transport of proton is facilitated? While the transport of electrons, how proton transport is across the membrane is explained in reason. Look at here first. In photosystem 2, when electrons is excited to P680, the primary electron acceptor is pheophytin. After electrons being accepted by pheophytin, it transfers to plastoquinone. Immediately plastoquinone absorbing the electrons, it will pick the protons from the stroma and it gets turned to PQH2, plastohydroquinone, and then it transports the electrons to plastocyanin and protons are pumped to lumen. Fine, right? So here we go. The primary electron acceptor transfers electrons to plastoquinone, enabling the proton translocation from stroma to lumen. Clear with quinone cycle? Okay. Accepting electrons, this will pick the protons from stroma. Protons are being delivered to the lumen and electrons are being delivered to cytochrome B6F complex. Cytochrome B6F complex. Clear, right? So yes, reason is true and it is correctly explaining the assertion. A and R are true and R is the correct explanation of assertion. Clear till here? And then next question. Identify the difference between C3 and C4 plants. C3 and C4 plants among the following characters. Photorespiration rate, true. C3 plants perform photorespiration, happens in C3 plants, whereas in C4 plants photorespiration is absent. So this is one of the difference. Activity of rubisco. In C3 plants it is in the mesophyll cells. In C4 plants rubisco activity is inside the bundle sheath cells. True. Type of light reactions. Light reactions are common both in C3 and C4 plants. Okay, which is non-cyclic photophosphorylation. Carbon dioxide primary acceptor from atmosphere. Primary acceptor in C3 plants is RUBP, primary acceptor in C4 plants is PEP enzyme, PEP, phosphoenol pyruvate. Anatomy of leaf. Leaf anatomy is dorsiventral, here also it is dorsiventral. So there is no point in that. And here the right answer will be anatomy of leaf. If you can consider cran's anatomy, then there is a difference. Both of them are dorsiventral leaves itself, but then bundle sheath cells possessing cran's anatomy in C4 plants is one of the variation again. So A, B, D and E, amongst which the correct answer will be A, D, E. If there is B and then you can also choose B. C is wrong. So you can't go with A, B, C options. The right answer will be D. Multi-statement based question, we need to choose wrong statements. Wrong statements should be chosen. Cytochrome C is attached to inner surface of the outer membrane. No, it is not attached to inner surface. Cytochrome C is attached to inner surface of inner membrane, not outer membrane. Fine. Electrons from complex 1 are transferred to ubiquinone in the inner membrane. True. This is from ETS. Temperature optimum for photosynthesis does not depend on habitat. It depends on the habitat. Those which are growing in the temperate regions are temperate plants, mostly C3 plants. Those which grow in the tropical regions are tropical plants, which are C4 plants. Temperature optimum varies based on the habitat where the plant is residing in. So this is wrong again. And then no linear relationship exists between light intensity and carbon dioxide fixation at low light. At low light, no linear relationship exists. It is at low light intensity, there is no increase in the photosynthetic activity. Only at high light intensities you can see the increase in the activity of the rate of photosynthesis. So this is also wrong. A, C, D are the wrong statements, where in this multi statements photorespiration, non-cyclic photophosphorylation and electron transport system Factors affecting photosynthesis. Two concepts are involved in this multi-statement based question. Option D will be the right answer. Next question you look into. Assertion reason type again. Look at here carefully, ma. When a green algae cladophora is illuminated in suspension of aerobic bacteria, bacteria accumulates in blue and red region of sunlight. Correct, because in blue and red regions only maximum amount of sunlight is being absorbed and maximum rate of photosynthesis happens, maximum oxygen evolution happens, and thereby aerobic bacteria got accumulated in these two regions. The bacteria release oxygen by the process of photosynthesis. Yes, by what type of photosynthesis has to be mentioned? It is oxygenic photosynthesis. So, as it is not specifying which type of photosynthesis, you cannot consider R as the correct statement. So, it will be A is true and R is false. Option C. Next question, again assertion reason type. Assertion. Electrons released from photosystem are used by electron transport chain during photophosphorylation. Fine, right? Electrons which are released from photosystem are used for the ETS during photophosphorylation. True only. Movement of electrons from pheophytene to PS1 reaction center involves oxidation reduction reactions only. Yes, it involves redox reactions, but nowhere it is explaining how the electrons are being utilized for photophosphorylation. Both the statements are correct, but there is no proper explanation, no accurate explanation for reason being explaining the assertion. Next, correct statements again. Photorespiration synthesizes neither sugar nor ATP during the release of oxygen. Yes. CAM metabolism is an alternative to C3 and C4 pathways of carbon dioxide fixation in dry climates. True. Bundle sheath cells have many chloroplastic walls and there are no intercellular spaces. After World War II, photosynthetic studies were exemplary. Yes, but then dark reactions were studied so far. Fine, right? So here you can go clearly with, as the statements are very clear, A, B, C. D is incomplete, so you cannot go with that. A, B, C are correct. Fine. Next. How many ATP and NADPH molecules are required for the synthesis of 6 molecules of glucose? For the synthesis of 6 molecules of glucose, how much amount of ATP and NADPH are required? Here we go. For 1 glucose formation, 6 Kelvin cycles will happen. 6 times CO2 is fixed and 18 ATP are formed and 12 NADPH, 18 ATP are required and 12 NADPH are required. Fine. For 6 molecules of glucose, 36 carbon dioxide molecules are required for fixation and 36 cycles will happen. 18 into 6 will give you the answer. And 18 into 12 will give you how much NADPH are required. Fine. So, the right answer will be option D. It is very simple. For single carbon dioxide, how much assimilatory power is required? For 1 glucose formation, how much is required? For 6 glucose, thereby you multiply it and it will, the assimilatory power will be compounded, and then you can look into the answer easily. Next. If 12 molecules of carbon dioxide are reduced, how many molecules of G3P are used for regeneration? How much of G3P are used for regeneration of RUBP in Kelvin cycle? Here you see 12 molecules of carbon dioxide are reduced. For 6 molecules of carbon dioxide being reduced, it is 12 G3P. For 12 carbon dioxide, it will be 24 G3P. Okay, it will be 24 glyceraldehyde 3-phosphate. Fine. If 12 molecules of carbon dioxide are reduced, how many molecules of G3P are used for regeneration of RUBP? For regeneration of RUBP, 24 molecules are present, okay, of which 4 will be transferred to glucose formation and 20 G3P for RUBP regeneration. Clear, right? So, here we go with how many are utilized? 20 G3P. Option B will be the right answer. Next question, which is match the following type of question. This is directly from factors affecting photosynthesis. Few of the options are missed. We will look into it again. Yeah, look at the last question of photosynthesis in higher plants. Identify the correct sequence of electron transport of photosynthesis. Correct sequence: P682 pheophytin, plastoquinone A, B, and then cytochrome B and cytochrome F, which is not followed by again plastoquinone. You can eliminate D. 100. Plastoquinone will not appear again. So, you can eliminate B as well. Cytochrome F will not be present in photosystem 1. So, you can eliminate this as well. Option D goes correct. Cytochrome B, cytochrome F complex. So, option C you see. Cytochrome B, F followed by plastocyanin, and then it is moved to P700. Option C will be the right answer. Clear? So, with this photosynthesis in higher plants is done. In session 2 we will address respiration in higher plants and plant growth and development. Clear right now? So, that is all for today's session. Clear till here? Any doubt anywhere? Is my voice clear now? Yeah. Next session we will look into the plant growth and development and respiration in plants.